Hello, I am Adriano Guzzi, and I am a professor of neuropathology, and I have spent most of my professional life working on prion diseases. And this is what I'm going to talk about to you about today. And this is a part of the Bio 6, sorry, just a moment, I'll share my screen. The Bio 615 course in biology for bachelor and master students. So what about prions? So what I will be talking about is I will compress into a typical lecture 3 centuries of prion sciences, and I will mostly follow the timeline that is depicted here, and that starts in 1732, when a disease was first reported in sheep, and it was called scrapie. And this disease is called scrapie because the sheep would scratch themselves and would eventually lose most of their fur. And then things did not really develop for a long period of time, but in the late 19th century, neuronal vacuolation was recognized as a feature of scrapie. And here you see a histology picture of a brain. This is actually from a human brain, from a person who developed a disease called Creutzfeldt-Jakob disease. And you see actually what I'm talking about, this vacuolation. It's also called spongiosis, because under the microscope, the brain looks like a sponge because of all these little vacuoles that have arisen. And then the scrapie was recognized as transmissible disease in 1936, with a very important experiment by 2 French researchers. And then I will talk about everything that happened afterwards. But let me also tell you that I have just published a review article in Science that reports about all of this, and you are very welcome to look at it if you are interested in more detail. So the first thread of the talk that I am giving will relate to a disease that is called Kuru. And this disease was extremely prevalent in the Fore population that inhabited the northern part of Papua New Guinea. And these people had the peculiarity that they had not encountered Western civilization for a very long time. In fact, they were shielded from Westerners until the 1940s, when missionaries started to go to Papua New Guinea, and you see it is very close to Australia, and people realized that there was a disease that was decimating this population. It was very frequent. It was actually the most common cause of death within this tribe called Fore. And but nobody really understood what was causing it, and there were all kinds of speculations. For example, some people thought that some kind of herb was causing the disease, that essentially that it was a poisoning, but there was no evidence for that. And other people even there were completely absurd theories. For example, there was one theory that this was a kind of a psychogenic hysteric reaction of Aborigines being suddenly confronted with Westerners. So all kinds of theories thrive, but the fact is that it was totally unclear what was going on. And then an American scientist, Carleton Gajdusek, went to Australia, and originally with the idea of working with Macfarlane Burnet, who was a famous Australian immunologist and a Nobel Prize winner who discovered the clonal selection of antibodies. But then Gajdusek actually found a colleague, Vincent Sigas, who was a doctor, and Zigas told him about this Kuru disease. And so Gajdusek decided to go actually and see for himself, and he visited Papua New Guinea, and then he stayed actually with these people for a very long period of time. And one of the things that, so he actually describes this disease, and in a series of papers published in the late 1950s, he described the histology, and the histology was on brain sections, on microscopical section of the brain from people who died of Kuru. He describes this spongiosis, this vacuolation that I was talking about before, and and in fact, this just shows you one of the victims of Kuru. You see that they are emaciated, and there was a severe neurological problems indicating that this was a brain disease. And this shows you what you would expect, this kind of vacuoles that appear in the brain. And then what happened was that there was a veterinary pathologist called Bill Hadlov, who used to work in Hamilton, Montana, and it's a pretty remote place. And Hadlov saw the description of Gajdusek, and he wrote a short letter to the Lancet indicating that, this looks very similar to scrapie. And as I told you before, scrapie is a disease of sheep that has been known for a very long time. And Hadlov said, well, we know that scrapie is transmissible, it's an infectious disease. So this Kuru thing, it looks very similar to scrapie under the microscope, so maybe it's transmissible as well. And this is really the experiment that Gajdusek then did together with his colleagues, Clarence Gibbs and Paul Brown, and they made tissue homogenates from the brain of people who died of Kuru, and then injected them into chimps, which is an experiment that nowadays would not be possible to do, because working on chimps is forbidden in most countries. And but they did this, and eventually what happened was that the chimps developed a neurological syndrome very similar to Kuru, and histologically it was the same picture. So this showed that Kuru is a transmissible disease, and for that Gajdusek received the Nobel Prize in 1976. And that established the infectious nature. But then the question was, how is disease being actually transmitted? And then Gajdusek found that the people, these 4 people in New Guinea, they were performing cannibalistic rituals. So when somebody died in their tribe, they would cook him and essentially eat him up. And this was not necessarily a belligerent gesture. It was more about honoring the dead. But what nobody really realized was that tragically they would disseminate the disease in this way. And because the problem is that the agent, which was much later was called the prion, and we will talk about this in detail, the prion is very resistant to heat and to all kinds of denaturations and disinfection methodologies. So as a result, Kuru became incredibly prevalent in the population. And now I want to talk about something. Let me show you something more about the vacuoles, which I think is very interesting. And this is an electron microscope picture that was given to me by Steve DeArmond that shows actually what these vacuoles consist of. So here you see the process, a neuronal process, then the black stuff is myelin, is the sheath of the neuron and of the neuronal process, and then you see that this is blown up, and then you have a vacuole. And here there is another one, and sometimes you can see some membranes, some degenerating organelles within the vacuoles. And this is really what the spongiosis is about. So these are neurons that are blown up. And still nowadays, this is extremely typical. If you see a vacuolating disease in a person with rapidly progressive dementia, then it's almost certain that this is a Creutzfeldt-Jakob disease, which is the equivalent of scrapie in humans. And to that effect, I wanted to make you understand also how did Creutzfeldt-Jakob disease come about? Well, Creutzfeldt-Jakob disease had been described actually even before Kuru. It was described at the beginning of the 20th century by two German physicians, Creutzfeldt and Jakob, who described this independently. And also Alzheimer had described one case that eventually turned out to be a case of Spongiform Encephalopathy, but Alzheimer thought it was Alzheimer's disease because it's very similar. When Creutzfeldt and Jakob described this disease, it was very, very similar to Kuru, but also Creutzfeldt-Jakob was not clear whether it was infectious. But then eventually Gajdusek has also transmitted Creutzfeldt-Jakob disease to chimps and shown that these are all infectious diseases. So let me now show you something more about prions, and this is much more recent. This is mad cow disease. Now, mad cow disease, if you are a millennial, you may actually have never heard of it, but in the 1990s, this was a major health scare, totally comparable to what is happening today with COVID. So what happened was the following. As I told you, sheep had scrapie. And, sheep and cows are herbivores. But then people were starting to say, it's why are we actually throwing away all the proteins that are contained in the offals? Because when you slaughter a cow, some parts of the cow go into for human consumption. You make the filet and the entrecote from that, but most of the cow is actually thrown away. So the idea, which was in principle a very green idea, was okay, why don't we actually take the offals and we recover the proteins, and then we can make food for the cows, so that we that there is a circle, and we don't circular economy, and we don't put to waste all this good protein. But then the problem is this ideologically may have been very attractive, but in reality what it did was that people then started to do the same thing with the sheep, not realizing that sheep very frequently have scrapie. So the scrapie agent was transmitted from one sheep to the other through this protein, and the problem was that the agent is very resistant, so you cannot essentially inactivate it, and it was also transmitted to the cows. And as a result, a lot of cows got infected. In fact, we can calculate that approximately 7% of all cows in the United Kingdom were infected with mad cow disease or bovine spongiform encephalopathy in the 1990s. And almost 300,000 cows died of this. But actually, the cows that died was not even the problem, because those cows were sick, so they were destroyed without going into the human food chain. But of course, a lot of cows were asymptomatic. So you see again the parallels to COVID. The asymptomatic spreaders, the super spreaders, were actually the problem. And here you see actually what happened in a timeline. So the first histopathological confirmation of mad cow disease happened in 1986. This was Wildsmith and Wells, two veterinarians in the United Kingdom, and then things picked up speed, and then the exponential growth in the number of cases. And then it was realized very quickly that the disease was being spread through the through the foodstuff, and then a feed ban was implemented. So people said, okay, fine, we now prohibit the use of these proteins for consumption to other cows, but you know, the cows that were infected still were getting sick, and the incubation time is very long. The incubation time is around 6 years, or 5 to 6 years, and you see that despite the food ban, the number of cases of cows developing the disease was getting higher and higher, and peaked only in 1992, and then it went down. So here you can actually calculate the incubation time. The incubation time is from here to here. And then the feed ban started being effective, and therefore now we have less and less cases. Now the problem was at that time, and you can see all the minimization of what you hear from politicians, and particularly American politicians these days, about COVID being not such a big problem and disappearing magically. You know, the same arguments were made in the 1990s, and this is a picture of John Gummer, the Minister of Agriculture in the United Kingdom, who was saying the same thing, was saying, you know, this BSE, this mad cow disease, it will go away, and there is no problem, and I'm eating a hamburger, and he is also, and British beef is safe, and you see, I give it also to my daughter, and then unfortunately it turned out that British beef was not safe at all, and here you can see what happened. So here you have the peak of mad cow disease, and then the disease was starting to disappear from the cows, but then a disease appeared in humans, because the humans were eating, of course, the disease meat, and then this disease called variant Creutzfeldt-Jakob disease, again, very similar to the disease described by Creutzfeldt and Jakob, but now affecting much earlier, much younger people, often teenagers, and then hundreds of people contracted this disease and died of it, a terrible disease. And you see, this is a tragedy that was brought about by human hubris, and which could have been avoided to a large extent if politicians would have listened to scientists. Now, what about the mechanism of transmission of prions? I will talk about this in the next installment. Stay on. Hi, I'm Adriano Guzzi, and welcome to the second installment of my short lecture cycle on the biology of prion diseases. So today I will be talking about the mechanism of transmission of prions. Let me share my screen, and here we go. So originally, everybody thought that prions would be some kind of viruses. After what I have described to you about Gajdusek and Kuru, everything pointed to a virus, because the agent obviously was filterable by a small pore filter, and so it couldn't be a bacteria, it was infectious, so what else could it be? So everybody has been looking for a virus, and in fact, Carleton Gajdusek himself started talking about something that he called slow viruses, which I think is a terrible name, because it led to a lot of confusion, because there are viruses called lentiviruses, and now I have to tell you that lentiviruses have nothing to do with prions, because the term lentivirus describes a subclass of retroviruses that take a long time to provoke disease, and that one, HIV is the most famous one. Now, slow means, obviously, lenti means slow, so that is the conclusion. But let me just tell you that the so-called slow viruses of Kuru and Creutzfeldt-Jakob disease have nothing to do with lentivirus, and anyway, they don't exist, because what was shown then afterwards was clearly that there is no inflammatory and no immunological reactions of the host that can be seen in the brain. And well, I would say that is perhaps a bit more too much of a politic statement, because in reality, what I'm saying is that you don't see in prion diseases, you don't see the kind of massive lymphocyte infiltration that you would see, for example, in herpes simplex encephalitis or in cytomegalovirus encephalitis. That does not happen in prion diseases. But what does happen is microglial, a lot of microglial activation and proliferation. Now, nobody has seen viral particles either, but I have to say that all these things are kind of circumstantial. The fact that you don't see viruses does not mean that they are not there. And as usual, absence of evidence is not the same as evidence of absence. However, one thing that people have seen was the deposition of some kind of abnormal protein, often in the form of plaques, and that's what you see here. And these plaques, you see them in Creutzfeldt-Jakob disease, you see them in Kuru as well, and Kuru, they are particularly prominent. Then there is Gerstmann-Streusler-Scheinker disease, which is a dominant, autosomal dominant prion disease that is very weird. It's something I will talk about later. And you see it also in scrapie and also in mice that have been infected with scrapie from sheep. And what you see is that these plaques are intimately associated with the spongiosis. So the vacuoles are in very close proximity to the deposition of these plaques. And this is now an immunohistochemistry. The brain has been stained with an antibody against this protein. What this protein is the prion protein, and I will explain to you in a moment what that actually is. Now, the important thing, and this was really the big discovery by Charles Weiss, the big discovery by Stanley Prusiner. By Stanley Prusiner in 1982, who published a very important paper showing that he purified these plaques, and he showed that the infectivity is contained in this plaque. But then a big surprise came, and the fact was that it was impossible to find any nucleic acids that would be associated with these plaques. Now, how can a virus possibly exist in the absence of nucleic acids? That again was a big mystery. And actually, there was even here I have to acknowledge another very important scientist, and that was Tikva Alpers, who was a radiobiologist, who showed before Prusiner that she could not inactivate the prion infectivity, the scrapie infectivity using X-rays. And now this is a very important point, because ionizing radiation damages nucleic acids. And in fact, it does so in a very controlled manner. And you can even infer the size of a genome by its X-ray inactivation spectrum, which means that if you have a very large virus, then you need relatively little radiation in order to damage it. And to sterilize it. If the genome of the virus is very small, you need much more X-rays. And now what Tikva Alpert found was that she could put as much radiation as she wanted, as she was never able to inactivate the scrapie infectivity. Now that was very surprising, and it also questioned, I mean, how can it possibly be that a virus can survive all this? And then Stanley Prusiner, in his famous paper in 1982, he proposed that the agent consists exclusively of a protein, that it does not contain nucleic acids. And that was, of course, something that most people were unprepared to believe. And Prusiner was really derided for this idea. But then, things started becoming a bit clearer. And now let me share the slide again. Here we go. Now, I have to say that the original idea of Prusiner was that the protein, that this plaque protein somehow would replicate itself, that it would be a self-replicating protein. And for that, Prusiner had actually harkened back to a suggestion by Griffith, a mathematician in the 1960s, who had predicted that such proteins might exist. But then what happened was that Stanley Prusiner was was at a meeting, I think it was in 1982 in Australia, and presented this idea. And Charles Weisman, who incidentally was my mentor in Zurich, was present at this meeting. And Weisman objected. He said, you know, if there is a protein, there must be a gene that encodes for it. There is no protein without a gene. And so Weisman embarked in an attempt to find the gene that would encode for the prion protein. And that was the work that he did together with his PhD student, Connie Basler, and then also Bruno Oesch. And they actually identify a gene. They identified the gene, and but that was there was a big surprise about that, because what happened was that they cloned the gene encoding for the plaque protein from hamsters that had been infected with scrapie prions. However, when then they did the control experiment, they looked for the gene in hamsters that were not infected, and lo and behold, there was a big surprise. The gene was present also in the non-infected hamster. So this doesn't make any sense. So if this is a viral, if this is a virus, it should only be existing in the infected hamsters. So how to make sense of all this? Now let me show you a couple of slides so that you better understand this. Right. So essentially, the gene encodes for PRP, what we call the prion protein, or PRP. And PRP is a protein that contains, well, it starts with some 240 amino acids, and it contains a glycophospholipid anchoring domain, and of course a signal peptide for directing it to the secretory pathway. So PRP is co-translationally extruded into the endoplasmic reticulum, and then it appears on the cell surface as a mature PRP that is anchored to the cell's membrane by a glycolipid anchor. And then the idea, and this is really how the prion hypothesis was modified by Charles Weisman, and the idea is that what really happens here is that you have a normal form that is called the PRPC, or cellular prion protein, and this normal form can misfold into something that we call PRP scrapie, or the scrapie associated isoform of the prion protein. And the idea is that PRP scrapie might be able to associate with PRP, and somehow instructing it to change its conformation such that it will also become PRP scrapie. And now we have two molecules of PRP scrapie that feed back into this circle, and then you have an exponential increase, because then from two molecules you will have 4, and then you will have 8 and 16, and so on and so forth. Beautiful hypothesis, compatible with the experimental data. Probably not entirely correct. The way we think of this process by now is more of a nucleation process. So what we think really happens is that PRPC and some kind of misfolded protein, which we may call PRP scrapie, they may be in an equilibrium with each other. However, this equilibrium is heavily shifted toward the left side. So whenever you have PRPC, you may have some small amounts of misfolded PRP. That is in and by itself innocuous, but the trouble starts when PRP scrapie aggregates, because now you have kind of oligomers, and these oligomers will grow very quickly, because they will recruit the PRP scrapie from the equilibrium, and then they will grow larger and larger. And at some point, you get real fibrils. So that's really how these things grow. That the oligomers elongate, and then they grow more or less like a crystal, and then they get longer and longer. At some point, they will break, and the breakage is actually the most important moment, because the breakage is when you go from one prion to several prions that will replicate independently. Now, this is how we think that prions replicate at the moment. But so how to prove or disprove this hypothesis? So Charles Weisman came up with a brilliant idea. And okay, I just want to repeat this. But there is one thing that I really want to tell you. So this is how we think that prions work. No? So you have the oligomers, and then the oligomer get longer, you have the fibrate, and the fibrate break, and then now you have prion replication. Now, the thing is that this phenomenon is not exclusively limited to prions. You see that something, if you take out the work PRP and you substitute it with A beta, then you have essentially the pathogenesis of Alzheimer's disease, because A beta does the same thing. It aggregates, make fibrates, and fibrates get longer, and so on and so forth. And if you take out A beta and you put synuclein, then you have the pathogenesis of Parkinson's and many other diseases. And by now, there is a long list of diseases, including amyloidosis, that obey to the same principle. So we are talking about something that started as a incident of nature, as a very rare disease, but now it's clear that it's pretty common. And the big surprise of all is that it even underlies some totally physiological mechanisms. For example, so what we are seeing here is a list of diseases. There are prion disease, there is Alzheimer's, there are tauopathies, so progressive supranuclear palsy and several other tauopathies. They all work in the same way, and the same applies to Parkinson's and to amyloidosis, and to some extent even to Huntington's disease, although their things are a little bit different, but the principle is the same. And but then, and this is really the big surprise, there are proteins that function entirely normal by doing the same things. And the first one was discovered by Reed Wickner, and it's in yeast, and it's a protein called SUP35, which is a component of the translational termination machinery, which is a complex of proteins that essentially takes apart the ribosome when the ribosome encounters a stop codon during translation. And that, of course, is very important, because if that doesn't work well, then the stop codon will be misread, and aberrant protein will be produced, which may be toxic. So it's very important that this works. And SUP35 works as well as a prion, and the prion property of SUP35 confer an evolutionary advantage to the yeast that are prion infected. And this was shown by Susan Lindquist, a fantastic molecular biologist who regrettably died a few years ago. And then other things were found. That Het-S does the same thing. The Het-S is a prion of filamentous fungi, and Het-S is really important for this reaction called heterokaryon incompatibility, which is very important for the mating of the fungi. And then also bacteria have prion-like molecules, and many other things. And I call these things prionoids because they are not really infectious. You know, the prions, they create disease, but the prionoids, they really don't do. Right. So that is what I wanted to say about this, but then I really want to go back now to the crucial experiment to prove and disprove the prion hypothesis, and this is what Charles Weisman has done, and his idea was the following. So if you think that the prion hypothesis may be true, then you would expect that if you take some normal prion protein and some pathological prion protein, so some PRPC and some PRP scrapie, then in essence, what Prosiner's hypothesis says is that PRP scrapie is capable of autocatalytically convert PRPC into more PRP scrapie, and this causes disease and replicates the agent. So and this was how things were standing in the late 80s, and then Charles Weisman, and this was in the beginning of the 1990s, so 30 years ago, came up with the idea, okay, what about if we destroy the gene that encodes for the normal prion protein, and so we make a knockout mouse, and then this knockout mouse will not contain the normal prion protein. And now if you try to infect it with a pathological prion protein, the mouse should not get sick. And so this was a crucial experiment. I should say that in those days, there were only very few knockout mice were made. Actually, only the beta-2 microglobulin knockout mouse was made by Rudi Jenisch at MIT. But essentially, this was a nascent technology. It was just at the beginning. And what was found is that mice that are knockout for PRPC completely resistant. They will never develop the disease. And if you look at their brain, there is no prion replication. So this was a very important experiment, and I'm very honored that that Charles Weisman offered me to help with this. And in fact, I did the pathology of these mice, and it was clear that there was no pathology. And this essentially, well, I would say, did not really prove the prion hypothesis, but it made it very, very likely. All right, we'll stop it here, and maybe we'll continue tomorrow.